Welcome to Week 5 of Hazardous Waste Treatment and Disposal. This week we examine abiotic and biotic transformation of hazardous wastes and processes of substance release and transport. Organic hazardous wastes can be transformed by abiotic or biotic processes. Abiotic refers to chemical and physical means. Biotic refers to biological means. Transformation reactions of organic contaminants involve the breaking of chemical bonds and the formation of new bonds. Metals cannot be transformed to another element, but metals can change oxidation state, which then significantly influences their transport in the environment or their behavior in treatment systems. The most important abiotic reactions include hydrolysis, photolysis, and oxidation reduction redox. Hydrolysis is the addition of water to a molecule and usually occurs at specific functional groups on organic molecules and is often enhanced by the presence of acids or bases. Photolysis is a light-induced redox reaction that has limited importance in the transformation of hazardous chemicals, generally because the chemical must be exposed to light for the reaction to happen. Redox reactions are promoted in the environment in which the chemical potential favors either the addition of oxygen or the loss of electrons or a hydrogen atom, or by the addition of electrons for reduction. Redox reactions are promoted by low concentration of oxygen-free radicals, by coupled reactions on the surface of minerals, or by a number of treatment processes such as ultraviolet light and ozone or electron beams. In biotic transformations, organic contaminants are metabolized by microorganisms in surface waters, soils, groundwater, and treatment systems through biochemical redox reactions. Microorganisms often use the contaminant as a carbon source and as an energy source. Of the common physicochemical and biochemical degradation mechanisms, biodegradation is the most significant process for the decomposition of contaminants in the natural environment. Biodegradation is mediated by microorganisms that are classified as decomposers. Their natural role is the metabolism of fats, proteins, carbohydrates, and other energy-rich biological compounds. These natural chemicals are synthesized by plants from carbon dioxide and energy of sunlight. Animals metabolize the natural molecules of plants and also synthesize some new ones. Decomposers degrade biological molecules to their thermodynamic endpoints of carbon dioxide and waste. This process of degradation, known as catabolism, yields energy which the organism uses for growth and mobility. Most organisms found in soils, groundwater, and hazardous waste treatment systems are unicellular and can be divided into two categories: prokaryotes and eukaryotes. Prokaryotes do not have a true nucleus, whereas eukaryotes have membrane-bound internal structures. The organisms responsible for the biodegradation of hazardous chemicals in soils, sludge, groundwater, and treatment facilities are primarily bacteria and fungi. Bacteria are the predominant form of biomass in soils, however fungi are common in acidic soils. Although protozoans, earthworms, and algae also decompose hazardous chemicals, their contribution to degradation is usually insignificant. Some common trends are found in both abiotic and biotic redox transformations. For both processes, the presence of oxygen determines whether oxidative or reductive processes occur. Obviously, the presence of oxygen will determine whether conditions favor the growth of anaerobic or aerobic microorganisms. In a similar manner, oxygen scavenges aqueous electrons in abiotic systems to form superoxide, which can form hydrogen peroxide. The obvious difference between abiotic and biotic transformations is the enzymatic catalysis that is inherent in biological processes. Reaction thermodynamics are relatively consistent for both abiotic and biotic transformations. For example, thermodynamics favor the reduction of oxidized contaminants like carbon tetrachloride, whether the transformation proceeds biotically or abiotically. In contrast, the transformation of reduced compounds like benzene proceeds most effectively via an oxidation process. In hazardous waste management, the assessment of contaminants after they are released from a source is often necessary. The source can be a wide variety of locations, like a contaminated site, an underground storage tank, a solvent recycling facility, or a landfill. The characterization of contaminants and their concentrations at the source and away from the source is necessary in order to assess risk, evaluate the need for remediation and treatment, devise health and safety plans, and comply with environmental laws. Contaminant release and transport dynamics can be evaluated through a number of procedures, each with a range of complexity. Contaminant distributions are commonly quantified using detailed sampling and analysis schemes as mentioned in an earlier week. Monitoring data are the most reliable, and actual data from the site or facility are preferred over modeling data. For a small site, a Phase 1 study may be conducted using only a conceptual analysis combined with a site visit, and no field data or calculations are used. More extensive site assessments and remedial investigation feasibility studies may involve relatively simple calculations or complicated groundwater and air dispersion models. The textbook covers the mathematical basis explaining the mass transfer of contaminants in the atmosphere and the subsurface, and the atmospheric transport following a volatilization release. The student is encouraged to review the math in the textbook. This last slide is a visual reminder of potential atmospheric and subsurface transport of contaminants after a release from a source. Whether the release came from an injection well, contaminated lagoon, land spreading, or other source, the flow path can be visually evaluated and considered.